Hello everyone, my name is Ivanka Tringovska. I'm a scientist at Marica Vegetable Crops Research Institute in Bulgaria. For many years I have been working with tomatoes, investigating their fruit quality and means to improve it. Now I'm very passionate about the phenotyping activities that we perform here at Marica Institute because I believe that knowing the biodiversity of a certain crop is a key for preserving this diversity and use it in breeding programs for creation of better varieties. In the next few videos, I will show some valuable tips about phenotyping of traits related to mature tomato fruits. I do hope these videos will help you in your own phenotyping activity. Now I am thrilled to be here today with you and can't wait to dive into the second part of the traits related to mature fruits of tomato. Let's get started. The first trait we will look at is fruit size. Tomatoes exhibit natural variation in fruit size due to a combination of genetic and environmental factors. Tomato size has been strongly selected during the domestication process, while tomato bears tiny fruits that are several hundred times smaller than the varieties we see today. Early humans cultivated and bred these wild tomatoes, selecting mutations associated with larger fruits and other desirable traits as taste. Over time, they developed larger fruits. The domestication process continued beyond the introduction of tomato in Europe. Farmers and breeders continued to selectively cultivate them to enhance desirable traits, one of which is fruit size. That's why now there is a big diversity in terms of tomato fruit size, from just 1 gram to more than 5 kilograms. In the CPVO technical protocol, the trait number is 26. It is quantitative characteristic and also a grouping characteristic. Grouping characteristics are those in which the documented state of expression can be used even if produced at different locations. The trait is denoted by asterisk, which means that it is important for the international harmonization of variety descriptions and should always be examined and included in the variety description. The recommended method of observation is visual assessment by a single observation of a group of plants. C in parentheses means that observations should be made of fruits at maturity. Here we have 9 states of expression given in an abbreviated scale, 1, 3, 5, 7, and 9. But note that all 9 states of expression should be used to describe the variety. So, between very small and small state, we have very small to small, which is 2, small to medium, which is 4, medium to large, which is 6, and large to very large, which is 8. Now, this is the theory. But how to phenotype this trait in practice? First of all, you have to phenotype this trait at full maturity state of the fruits, since tomatoes continue to gain weight even after they have been harvested. So, don't judge this trait on green or non-well matured fruits. Wait until full ripening state and evaluate fruits from at least 20 plants. Do your assessment preferably on the second truss or higher and avoid first and last trusses. Since this trait has to be evaluated visually on a group of plants, you will need to determine the average image. To do so, leave aside the largest and the smallest fruits of the sample. These are usually the first and the last fruit from the truss, and then compare with the example varieties to decide on the proper nodes. An alternative evaluation of this trait is by measurement. Although tomato fruit size and weight are two distinct characteristics, the size of tomato is often judged by its weight. Measurement is done by weighting 20 average fruits per sample, and again, the largest and the smallest fruits have to be left out. Weigh preferably fruits from the second truss or higher and compare with the example varieties. In our Harnestone phenotyping trial, we are going to assess this trait in a slightly different way. We are going to weight individually all fruits from first to fifth truss or for indeterminate accessions and all fruits for determinate ones. This will allow us to examine precisely not only fruit size but also traits as yield, fruit set, and fruit set homogeneity. Let's move to the next trait, which is thickness of the pericarp. The pericarp is the outer layer of tomato fruit that surrounds the seeds and refers to the fleshy part of the fruit. Its thickness is important, for example, for the processing industry, where thicker pericarp is preferable because it results in a higher yield of tomato paste or sauce per unit weight of tomatoes. In the CPVO technical protocol, the trait number is 35. Similar to trait fruit size, pericarp thickness is quantitative characteristic which has to be assessed at full maturity state of the fruits. The recommended method of observation is visual assessment by a single observation of a group of plants. Under CPVO number column, we see a plus in parentheses. This makes reference further in the document where it says that the thickness of the pericarp has to be observed as an absolute characteristic, irrespective of the size of the fruits. Again, here we have 9 states of expression given in an abbreviated scale, but all 9 states of expression should be used to describe the variety. To phenotype this trait in practice, you have to take mature fruits from at least 20 plants. Take out the largest and the smallest fruits in the sample that are usually the first and the last fruit from the truss. Cut the center of the fruits horizontally and observe the absolute thickness of the pericarp. Don't take into account the fruit size and compare with the example varieties to decide on the proper state. In our Harnestone phenotyping trial, we are going to assess this trait in a slightly different way. We are going to use one very good and precise tool for assessment of this trait, which is the Tomato Analyzer software. To use it, we will need to scan horizontally cut fruits at high resolution, usually 300 dpi. We continue with trait number 36 from the CPVO technical protocol, which is number of locules. Locules are the compartments that contain the seeds. Their number is determined by the number of carpels in the flower. Wild species of tomato have fruit with 2 to 4 locules. Cultivated varieties that contain the mutant FAS allele can develop more than 15 locules, which increase the fruit size. In other words, the number of locules is connected with the fruit weight, and the increase in locule number is an important step in the development of larger tomato fruit. It is also a trait altered strongly by domestication process and breeding of tomato crop. In the CPVO protocol, the symbols in the first 3 columns tell us that the trait is important and should always be examined and included in the variety description. It is quantitative characteristic that should be assessed at maturity. Here we have 2 alternative methods of observation. One is visual assessment by a single observation of a group of plants, and the other is measurement of a number of individual plants. Here we have 9 states of expression from 1 to 5 that corresponds of 2 to more than 6 locules. The steps of assessment of this trait are similar to those of pericarp thickness. You will need fruits from at least 20 plants with representative shape and size. Take out the largest and the smallest fruits in the sample. Cut the center of the fruits horizontally and count the number of locules. In case that you have a variety with 2, 3, and 4 locules and you have doubts about the state 2 or 3, cut more fruits to see the leading expression. In our Harnestone phenotyping trial, we are going to assess this trait as I just described. Those were the first 3 traits. I do hope you understand them better now. I left 4 more traits for the next video. Now feel free to leave any questions or comments in the chat. I'd love to hear your thoughts and experiences. Thank you for watching and see you again in a while.